Welcome all to Monica's Foundation classes. Today we are going to discuss in brief about the subject epigenetics. Epi means above. Genetics means our genetic code, our genome. Means something that is above our genome is called as epigenetics. First, when we talk about genome, what is our genome? Like humans, we are diploid. That is, we contain 46 chromosomes. Out of, if we consider one pair, we have 23 and 23. That those 23 are again duplicated, to give us these 46 chromosomes. So whenever we talk about our genome, we talk about this only one set of chromosome. Our one set of chromosome is our whole genome. Like in scientific study, whenever we want to study our whole DNA, so we will study about our genome only. You know why? To duplicate our study. To first we will study this first strand, then again the same amount of DNA. So whenever we talk about epigenetics, we generally talk about our genome. That is one set of our chromosome, including X, Y, and all our 22 chromosomes, autosomes. Okay. So now considering the topic of epigenetics, now epigenetics is a broad subject to study in which we consider the chemical epigenetics usually refers to the term to chemical modifications that is above our genome. So consider this is our genome, and in epigenetics these are epigenetical chemical modifications which takes place above our genome. So this we can consider like it is a soap and water is floating above it. So our soap is our genome and the water is our epigenome, that is genome with epigenetic tags. What are epigenetic tags? These are nothing. Epigenetic tags are just the name given to the only chemical modifications. Like see, this is our soap and this is our DNA or genome. The DNA or genome is the same thing. Okay, so this is our DNA, and above the soap the water is floating, and this is our epigenetic tags. Now talking about epigenetic tags, that how this water, that is our epigenetic tags, that change the shape of our soap, that is DNA. Now, this is the whole study about epigenetics subject. Okay. Now, considering first making the shape of our soap, that is our DNA, then uh, talking about epigenetic tags. Now, epigenetic tags are only the chemical modifications. These are chemical modifications which include methylation, that is the attachment of methyl tags. CH3 is the chemical formula of methyl. Okay, attaching of methyl tags, second OCH3, acetyl tags, that is acetylation. Tags, what is tags? Like our clothes also have tags, somewhat our DNA also has tags. Okay, so acetyl tags are just the chemical compounds, molecules, chemical molecules which attach above our DNA, like methyl tags, acetyl tags. And third is phosphorylation, means phosphate tags. And fourth is sulfonation, sulfate molecules attach on DNA. Now these are so many modifications depending upon which chemical attaches on the DNA. Now talking about methylation, whenever the methyl CH3 molecules attaches above the DNA, usually it leads to repression of genes. That is, genes do not express that particular gene part. For example, encoding your black hair, if that gets methylated, then that your particular black hair gene would not express and you will ultimately have white hairs. Okay, now second, considering acetyl tags, these usually upregulate your genes. That is, your genes get expressed at much higher level. Your expression of genes increases. For example, if your gene encoding a black hair is having acetyl tags, then your black hair will be more blackened, more darkened, and you will have more black hairs. So that what acetyl tags do. If acetyl molecule gets attaches over your DNA, then you will have more black hairs, okay, and more amount of hairs. This is only a hypothetical example, it is not a real one, but for your clear understanding only I am giving this example. Now, considering phosphoryl and sulfate tags, these are neutral. These can somewhat decrease expression or increase expression. These all also can change their major part, um, only under some conditions. Like methylation, its major part is to reduce the gene expression, but in some cases it can increase also. And same with acetyl tags, mostly it increases gene expression, but in some cases it can decrease also. Now, if we talk in detail about how they decrease or increase, it is somewhat basic concept. Like, this is our DNA, and here comes the promoter is that site where our DNA polymerase or RNA polymerase attaches, and it duplicates the strand of DNA by moving along it. Okay, so if suppose this is our DNA, okay, and mostly uh, this is our promoter site, that is 80 rich, means it is having more of 80 sequences. We will draw again to give you clear understanding. Suppose this is our 80 rich region having many 80s, okay, and this site we consider as a promoter. It is generally giving name name as promoter because it is the only site where the DNA polymerases attach and promotes our DNA replication, duplicates our DNA. So if in this promoter site, if we have methyl tags, so it would be very difficult for DNA polymerase to attach here. If DNA polymerase will not attach here, therefore our DNA will not be get duplicated, and our duplication and our DNA will not be replicated, and a duplicated strand will not be made. So, on contrary, if on promoter side we have acetyl molecules, okay, so these acetyl molecules will promote. DNA polymerase has such structure that CH3, it can't bind to CH3 molecule with such affinity as it could bind to COCH3. What it has only CO in addition to CH3. But DNA polymerase has such affinity to bind to COCH3 that it will bind so strongly to the promoter with great affinity that our DNA strand will get duplicated and will result in replication of our DNA. So the more the same gene, this is our whole DNA, and we call that region a gene which codes for a useful protein in our body. For example, this protein could result in our black hair, or it could increase our albumin content in our it could increase our albumin content. And DNA polymerase is also a protein, okay. It was first encoded by a gene, and this DNA polymerase was made, and it again act upon the gene, okay. So this is whole thing about epigenetics. It is a broad study of how some chemical modifications or chemical tags, which are like a liquid on a soap, how they modify our soap, that is our genome. Thank you, students. If you like this video, please subscribe to Monica's Foundation classes. Thank you.